All right, everybody. We are going to look at natural selection today. So, when we're thinking about natural selection, a lot of times we think of survival of the fittest and how it differentiates from artificial selection. So, in artificial selection, we have um, organisms that are chosen by people because of certain traits, and we're basically shaping what we want in the gene pool by selecting characteristics from the phenotypes of whatever we're domesticating. But in natural selection, we're taking a look at more what natural characteristics are beneficial and will increase survival. And if it increases survival, we think that it's typically going to increase their ability to reproduce as well. So some of these characteristics or adaptations that come along may help them, gather more food or be what we would think of as dominant. But a lot of times it's just helping them survive maybe diseases, and they have better ability to fight off different types of things that could kill them. So, natural selection. Again, this is getting into Darwin, and we're going to break down natural selection into four distinct parts. And going back to Darwin's theory, he noticed that individuals are different, and he said that variation is inherited. So, it's not individual or populations don't change over time because they learn new things or because they stretch their necks or anything like that. It has to do with um, population change over time because it's passed on from generation to generation. So there are certain traits that are not passed on from generation to generation. For example, if I learn a lot about algebra, my kids don't automatically know algebra. Okay. So another big key point is that more offspring are born than can survive, and that's going to lead to competition for limited resources. And again, a lot of times we think of competition being like one thing's fighting the other, and that's not necessarily the case. Um, as we keep on going on here, this variation is going to have some be at an advantage and some are going to be at a disadvantage. And again, getting back to this idea of fitness, it's reproductive success. It's not like how fast can you run a mile. Eventually, those that are best suited, that have the advantages in their phenotype, are going to be able to reproduce, and they're probably going to pass on those traits in some way to their offspring. And then species change over time because of that. So, moving on, our stages here, we're going to look at overpopulation. More offspring are born than can survive. Those that are born are going to be different from one another. We're going to talk about mutations and gene shuffling a little bit more in depth here. They're going to struggle to survive. If they're in the same niche, they're going to compete for, the same habitats. They're going to compete for the same resources as far as food, water, shelter. Um, so if they occupy the same niche, it's not just the same habitat, but it's the same role in the food web as well. And finally, survival of the fittest. Those that are best suited will pass on those genes to their next generation. So this little simplified little cartoon version here. So you got mama spider here and having lots of babies, more than what are needed to replace the previous generation. All of those little baby spiders are going to be slightly different from one another. And all of those different variations are going to be in some ways helpful and in some ways harmful. So if one of those offspring had this ability to like be jumping, jump amazingly well and be able to capture more food, if it could capture more food, maybe it could live longer and be able to pass on the jumping gene to its offspring. And if it is able to have an adaptation that is more successful, you would expect that adaptation to be passed through genes, and more offspring would have that characteristic as well. So then ultimately, over time, you might have a little shift in the way the population looks or acts, um, in a with particular traits. So, number one, let's go in a little bit more depth here. Getting back to ecology, and again, evolution is often thought of as just ecology over a longer period of time. So, if you're tracking populations, you can see that their population fluctuates. And when we were studying ecology, we learned about K-selected and R-selected species. And K-selected species, again, a lot of times have fewer offspring, but take really good care of them. And but at the same time, they still go over the carrying capacity. They have more offspring than can survive. So they're they're once you're above carrying capacity, those that are alive are competing for limited resources, and it's going to fluctuate closely around the carrying capacity. Whereas our selected species have a tremendous amount of offspring, oftentimes don't take very good care of them, but this is the boom and bust cycle of our selected species. And you have this limit to what the habitat can maintain. And when it's above that limit, only the most best adapted to the environment and to survival are going to be passing on their genes to the next generation. So, overpopulation, there's too many babies. Now, I know I kind of simplify this with too many babies, different babies, the babies compete, and the best babies will live to reproduce. But we're going to go a little bit more in depth here, and some of these things are very common to us because we've been talking about them for a while, but some are a little bit new. So we have different we have offspring with different characteristics, and that comes about through mutations in gene shuffling. And just thinking about humans and Punnett squares and diploid possibilities, uh, if you took the amount of chromosomes we had and independently assort them and then randomly fertilize, you could really end up with 64 trillion diploid combinations, which is pretty phenomenal. Now, does that mean that they're going to have all different alleles, 64 trillion different alleles? Not necessarily, but you can have so many different combinations. It's incredible, and that's what sex does. So when we're talking about mutations, we've got point frame shift, and point mutations are just a substitution, and that's only going to change one amino acid typically, which could cause a slight change in shape. It might not necessarily have a great fluctuation on how that protein functions, but you're gonna have a slight change. Frame shift though, with an insertion or deletion, can often have potentially very destructive effects, and it's going to change everything following that. So you're really going to get a protein that has a very different structure, which may ultimately cause it to be a new gene. And when we start thinking about genes, everything about variation, all of the other things only shuffle around the genes that exist. And mutations are really the only way that we get truly new genes formed. And new genes come about through the evolutionary process. So we can have mutations. And a lot of times, this is the mechanism by which they can change the letters of the DNA code. But a lot of times it's not always about making new genes, it's simply changing the expressions of the genes that already exist. Because you can mutate things that not only cause new proteins to be made, but it changes how they get turned on or how they get turned off. And if you express a gene for a different amount of time or during a different stage of development, you're going to have a very different phenotype possibly. Okay, so again, point mutations, you change one letter, you change one amino acid, unless it's in a stop codon, and then you can have different things. But even this little thing, its little substitution doesn't really change the protein very much. Well, it can be enough to like change the hemoglobin protein enough to make it sticky, which then causes sickle cell, which again can be good and can be bad. The disease is bad, but being heterozygous is actually an advantage. So when we start talking about big frame shift mutations or chromosomal mutations where we can actually, with the chromosomes, we can actually break things off and have them not reattach, and you can lose things or add things on to different chromosomes on a huge amount. But we can also look at insertions and deletions in a single letter format. And if you add something or you delete something, you're going to change the reading frame of the triplet codons, which then could possibly change every single amino acid after that, which could be huge. Now, when we start talking about gene shuffling, usually we think of sex. Okay, so we have creation of gametes, we have sex cells, we have egg, we have sperm. And part of meiosis is that there's going to be crossing over, and they're going to swap parts of the chromosomes. In addition, there's independent assortment. Which one lines up on the left or the right from during metaphase is random. Okay, so you can have different combinations of alleles put together, which creates different haploids, so different possible egg and sperm. And then random fertilization, depending on which sperm or which egg actually come together, we could have, this is where our 64 trillion comes up. So this is like 2 to the 23rd possible combinations, and this is 2 to the 23rd possible combinations. When you cross up those, you could have, just phenomenal amounts of variety. And again, we have crossing over here during tetrad formation of metaphase 1 during meiosis 1. So we've got that can introduce new allele combinations, but even this, so independently assortment, we can have all of these different possible varieties of sperm or egg. And then the zygotes, so meiosis produces gametes, and then fertilization produces zygotes, and we could have a tremendous amount of variety here. And every time we add a new trait, we ultimately have the possibility of creating, of exponentially growing a Punnett square. So now this is a little bit new getting into prokaryotes. How do they shuffle their genes? Because bacteria can be different from one another. However, they're not really having sex. So we just talked about how amazing sex is at creating variety, but the thing is, bacteria and prokaryotes can have variety besides just mutation as well. Mutation is a major part. However, they can exchange DNA with each other as well, or with the environment. So we have conjugation, which is the closest thing to, bacterial sex, and they transfer some plasmids. So we've got chromosome, the single chromosome, but then you also have little plasmids in there. And those plasmids can also be taken up from the environment in transformation, which we'll look at with our bacterial transformation lab. And you can also have transduction, where viruses actually infect and destroy bacteriophages. Viruses, tear apart a bacteria and steal some of its DNA, and then actually put that DNA into a different bacteria, and in that way they can transfer DNA from one to another. So conjugation, this is what I mentioned as the closest thing to bacterial sex. And when this happens, again, they have to be in the right, have all the right components here. But you can have these little pilus or pili that extend from the outside of a bacteria. And these little branches that hang off can actually sometimes attach to other bacteria. And when that happens, it's possible to replicate plasmid and transfer the DNA within those plasmids to another bacteria. Staying on this idea of plasmids, bacteria can also, under the right conditions, accept plasmids from the environment, and then it's gaining DNA information, and this is creating variety. The final way we're looking at here is transduction. So we've got this virus, right? And we've got a bacteria. And you've got this bacterial DNA here, and you also have DNA from the virus. And this virus is going to inject its DNA into the bacteria, which then would lyse up all of the DNA there. And ultimately, as it has these enzymes which break down and infect this cell, everything's going to get chopped up together basically. And now it's going to hijack the machinery of that bacteria to ultimately use its ribosomes to recreate the protein coat for all of these viruses. But sometimes in this process, it doesn't just repackage its own DNA. Sometimes some of the DNA from the original bacteria can slip into these bacteriophages. And when they get big enough and burst out of this cell and destroy this cell, this virus is now transferring some of this original bacteria's DNA, and then it can go and infect a new bacteria. And as it does that, maybe it doesn't have enough of those lysing enzymes to fully destroy the next one, but it's still injecting its the original host bacteria DNA into this new host, and that can get inserted into the DNA structure of this new bacteria, which then adds variety to the new bacteria. And that's transduction. One thing I want to mention really quick is this idea of transposable elements, just because it's so incredibly interesting. So another idea of gene shuffling, when you think of shuffling, you think of mixing the deck often, and that's definitely occurring when we start thinking about sex. What gets put together and shuffled together in which orders is a little bit random. Um, and you also have random kind of things happening within the DNA. And these transposable elements, you can actually take pieces from the large bacterial chromosome, take those pieces out and insert it into a plasmid. So it's transposed DNA from here to here. And now we just talked about all the different ways plasmids can be transferred, either picked up from the environment or transferred through pili. So now we can actually create variety in that way. And this transposable elements is kind of a big deal when it comes to eukaryotes. And it's kind of thought that some of this might be the basis of retroviruses or how viruses work as well, because we it's pretty amazing, and we don't often teach this in biology. But if you have your genome, your DNA, you can you make your normal messenger RNA, you turn on a gene or you turn on a piece, and it comes out here. We transcribe a section of this. This transcript can now be picked up by this enzyme, which turns it back into DNA, reverse transcriptase. So now it's going to get turned back into DNA, but now this DNA, which is a copy of this original part, can now get stuck in to different areas of our DNA. So when we start thinking about like, okay, we have 20,000 genes or 25,000 genes, but we have so much what we would call junk or extra DNA within there. And some scientists hypothesize that, some of that, all that extra stuff is coming from these retrotransposons. And they're taking these things and they're putting them into different spots, and that makes the DNA longer and longer and longer and longer. But that doesn't necessarily, this might not actually be what we consider a gene for a protein. It can just be an extra piece of the DNA that can be put into other parts. Now, why could this be really significant? Because this part that is taken out here could possibly be a promoter sequence. And that promoter sequence is what turns on genes. And this promoter sequence could possibly have a lot of activators that that make that gene get made a lot, make a lot of that protein. And if you take this activator sequence and you insert it in front of another gene that maybe doesn't get turned on very often or isn't meant to be turned on very often, and all of a sudden you're going to turn on that gene and make that protein during a different time in its development, um, or it just might make it in a new type of cell, which this is a big part of cancer research these days because proto-oncogenes um, aren't are cancer genes in a way, but they're like the gas pedal pushing cell division. And as long as they aren't, you only make those proteins that push cell division at the right time, it's not a big deal. However, if you translocate or transposition a new promoter in front of this proto-oncogene, now you're going to be making this growth, stimulating this type of protein that pushes cell division way more. So you're going to insert this really active promoter sequence in front of something that pushes cell division, and then all of a sudden, when a gene should be turned off and be in a G0 phase, all of a sudden it starts getting the gas pedal stuck and pushing it through cell division and pushing it through cell division, then we have cancer. So again, some of this kind of gene shuffling stuff is really exciting because as we learn more, we understand how it affects disease and everything like that as well. All right, so finishing up quickly here. The third step in natural selection is a struggle to survive. So when we're above carrying capacity, there is going to be a struggle right here. And, I kind of stick with this funny little babies stuff, and babies compete. Well, rarely do babies actually compete. There is this thing called obligate siblicide, where Cain and Abel effect, where they two eggs are laid and only one will be cared for, and whichever um, baby chick pushes the other one out of the nest ends up getting all the attention, and the one that gets pushed out of the nest, the parents just ignore it and let it die. So there is like baby competition for survival, but it's rare. Basically, when we're thinking about bringing us back to ecology here, there's going to be density dependent limiting factors that are going to affect who survives and who dies once you're above carrying capacity. And these, what carrying capacity is dependent on food, water, shelter, sunlight, predation, disease, all of these density dependent limiting factors. So if there's more food, the carrying capacity might go up, and then there's a little bit less competition. However, in times when maybe there's a low amount of water, you're going to have more competition, and that might change the habitat. And in drought conditions, certain phenotypes might have more of an advantage. They might have better survival adaptations to drought conditions, and then you're going to change the allele frequency, or you're going to slightly change the population. Okay, finally, survival of the fittest. Again, fitness does not indicate strength or size. Being big isn't always good. So it's not always about like strength or size. Fitness is only a measure of reproductive success. So if something is fit, that means it's able to pass on its genes easily. So when we're thinking about these moths here, the classic moth study, um, these dark colored moths in this environment are more fit because they are more likely to survive and more likely to pass their traits on to the next generation. So in a dark environment, you would expect this frequency of the allele to increase, but in a light environment where these would blend in, you would expect them to have better reproductive success and be more fit. And again, we start looking at this almost like an ecology standpoint. We start looking at populations and how the populations change over time, and that's the basis of evolution. So if we're looking at these plants and you have the original population, if having more spines on the plant is an advantage to reproductive success because they're not getting eaten, you would see a shift in the population, and basically that's evolution. through natural selection. Something changes in the phenotype and they're better adapted to reproduce. So again, stages: overpopulation, inherited variation, struggle to survive, and survival of the fittest. And that is the basis of natural selection. You got to have too many, got to be different. Those different ones are going to compete for limited resources. Some are going to be better adapted than others, and those that are best adapted for the environment are going to pass on those genes to the next generation. All right, so that's natural selection.